So let's go ahead and look at the actual tissues that are going to make up the organs of the body, which remember is that tissues are clusters of cells that will have a specific function for the bodies themselves, and we'll find tissues throughout various types of organs within the body. We have four principal types of cells. We have epithelial cells, muscle, connective, and nervous in terms of the tissue types. And so let's go ahead and take a look at the epithelials. So we'll start with some general characteristics of what epithelial tissue happens to be in terms of the epithelial cells. They're going to be found on the surface of everything. So any type of organ, any type of other tissues will be covered by epithelial tissues. These are going to be the principal cells that we'll see within the epidermis in terms of the integument. They're going to be characterized by having cells that are closely packed together. They'll be connected via tight and desmotic junctions. The principal types of desmotic junctions that we'll see will be not true desmosomes, but more of a hemidesmotic junction in terms of the lines around the organs. However, when we get to the epidermis, we'll see them more as a desmotic junction as we get into the upper stratas. When we talk about the epithelial tissues, we describe them based off of either their shapes being flat or squamous, cubed or cuboidal, and then rectangular or column-shaped or columnar. We then differentiate them based off of their layering. And so within the epithelial tissues, we will see distinct layerings of cells. If we have a single layer of cells, we usually refer to it as being simple. If we have more than one layer of cells, we start getting into stratified. Now there's another type of classification that we have within this stratification, where if all of the cells are of the same shape and the appearance of the tissue has a stratification to it, but all of the cells come from the basal membrane, then we refer to it as being pseudostratified. The other type of the stratification network lies in what's referred to as transitional. And within transitional, what we have is we have layering of the epithelial cells, but we don't have a clear, distinct delineation as to what actual cell type we are looking at. And so when we start looking at some of the histology images and looking at the slides of the tissues, what you're going to be starting to look at is these distinct lineages of cells based off of their stratifications and their shapes. And so we'll look at some of these in more detail when we look at the actual histology slides. The last type of specialized epithelial cells that we have to look at are things that are referred to as ciliated and microvillate. The ciliated and microvillate epithelial cells are principally going to be lining lumens of the body, and they're there to move stuff across or to increase the surface area of exchange like we talked about before when we looked at the membranes of the cell. Then there are specialized secretory cells, which are referred to as goblet cells, and we'll take a look at specific goblet cells within the trachea that are going to be producing viscous secretions that form the medium within the lumens. In the case of the goblet cells within the trachea, it's going to be forming the mucus that we see within the respiratory tract. Then we have other specialized secretory epithelial cells, which are referred to as islet cells. And what islet cells are is they're clusters of cuboidal cells within a larger glandular tissue that are separate from the rest of the glandular tissue within a special type of epithelial cells known as glandular epithelium. So let's take a look at what we're talking about when we talk about this connectivity between the cells. So if we have just a single layer, so if you notice at the top of the slide, there's a row of cells that are all connected to the basal membrane. So the basal membrane is the set of other tissues, connective tissue in particular, that the epithelial cells are going to be connecting with. If we have just a single layer of cells coming off of that, we have what's referred to as simple. Now, if we have multiple layers of cells coming off of that basal membrane, we now get what's referred to as the stratified. Now, if we look to the lower corner, we notice that we have different cells based off of the shape. If we have a more of a flattened cell arrangement in terms of its cytoskeletal structure, we get what's referred to as the squamous. The squamous is going to be there for cells where we have to have large amounts of diffusion heading across the cell, or where we don't want the cell to have a large amount of volume to it. We want them to be excessively closely stacked to each other, like what we see within the epidermis of the skin. Then we have the cubed shape, or the cuboidal. The cube shaped and cuboidal we'll see with cells that are going to be involved with absorbing large amounts of material or excreting some materials. And then we get the columnar, or the rectangular shaped. And just like the cuboidal, they're going to be involved with the secretions and absorptions of materials. And so we'll see these cuboidals and these columnars within and around lumens, where we're going to be absorbing things like within the intestines, or excreting things like what we see within the nephrons. The other type of arrangements that we see within epithelial cells is what's referred to as the glandular epithelium. And so glandular epithelium is epithelial cells that are predominantly secretory cells. That is, they are cells that are going to be secreting chemicals into other things, or secreting chemicals onto, say, the body itself, in terms of stuff that goes onto the skin, into the integument, and on top of the epidermis. When we look at the glandular epithelium, we talk about it based off of either the structure that the gland takes, in which we have 8 general structural characteristics, and we'll take a look at each one of those here in a second. Or we talk about it based off of the mode of secretion. In this case here, we have 3 types of general secretions, and once again, we'll take a look at those 3 general types. So we'll start with the structures. And so the most simple of these secretory structures, or glandular structures, is what's referred to as the simple tubular. So if you notice in the simple tubular, what we have is we have a tube, or a column of secretory cells at the base of the duct, surrounded by what's referred to as the duct region of the gland. We see these types of epithelial tissues in the intestines. So intestinal glands will be made up of this simple tubular glandular structure. The next most complex is going to be what's referred to as the simple branched tubular. And so the simple branched tubular, what we have is we have tubes again, but instead of having a single tube, we now have multiple tubes. And these multiple tubes are going to junction together into a single duct. An example of this is a merocrine sudoriferous gland. And so these are going to be the sudoriferous glands that are responsible for not producing the sweat that we think about when we think about sweat, but these are going to be the glands that are going to be more involved with the production of other substances that we see in sweat beyond just the liquidy sweat that you might be thinking about. The next is the simple coiled tubular. And so once again, what we now have this extended simple tube. But because it's running out of space, it starts to coil around itself. This simple coiled tubular structure is what we see when we actually look at the apocrine sudoriferous glands, or what we think about when we think about actual sweat glands. We now get to the second type of secretory structure, and that is the acinar. And so in the acinar, what we have is we have a small sphere of secretory cells connecting to the duct. You can think of this structure kind of like a cul-de-sac, in which the duct kind of heads down and all of a sudden it comes into this loop at the bottom of the duct, where there's a longer duct and a shorter secretory region than what we see with the tubulars. In the acinars, we'll see this simple in gastric pits, as well as the mucus glands of the gastrointestinal system, what we usually will see within the esophagus and within the duodenum. Just like we had with the tubular, we will have branched within the aceners. And so we have the simple branched acener. And the most common of the simple branched acener is the sebaceous glands. And so what ends up happening with the simple branched acener is that we have multiple acener spheres connecting together into a single duct. And so that's the end of the simples. We now get to what's referred to as the compounds. And so the compounds is where we have multiple ducts heading into a single pleural ductal opening. And at the end of each of the ducts, we will either have tubular regions, where we have multiple tubes coming onto multiple ducts, which then head into a single duct. We see these types of epithelial glandular tissues in the salivary glands, as well as in the male reproductive glands, in particular within the prostate gland and within the seminal vesicle. The next type is the compound acener. And in the compound acener, what we're doing is we're exchanging the tubular part for an acener part at the end of all of this branching networks. We see these within the sinusoidal lobules of the mammary glands. The last of the structures is what's referred to as the compound tubuloacener. And so what we've now done is we've now taken and we've elongated these acener regions. And by elongating these acener regions, what we've done is we've added a little bit of a tube to it. And so since we now have a tube part on the edge of the acener part, we now have both a tube part and an acener part within the gland. We see these within the salivary glands, as well as within the acener cells of the pancreas. So those are the structures of glandular epithelium, but what about the secretory parts? Well, there's three types. The first type is referred to as the merocrine. And so the merocrine is going to be cells where we'll be producing chemical secretions from those cells that will be exocytotically released into the duct to be secreted. The next type is what's referred to as the holocrine. And so in the holocrine, what we have a group of cells on the basal membrane of the gland that's undergoing cellular division. As it undergoes cellular division, it starts to propel itself towards the duct. And as it starts to propel itself towards the duct, the distance between the cell and the basal membrane becomes so great that the cell cannot metabolically stay intact. And what it then does is it then disintegrates. And as it disintegrates, it starts to break apart. And what is released from the holocrine is this small bit of cell with all of the debris within the cell being secreted. The last type is what's referred to as the apocrine. And so in the apocrine, what we have is we have a rapidly dividing cell that instead of undergoing mitosis, pinches itself off and then releases that pinched bit of cell into the lumen of the tube, which will then go ahead and be secreted from the gland itself. And so let's take a quick look animation-wise as to how these three glands will function. So we have our merocrine, our apocrine, and our holocrine. So notice that with the merocrine, we are simply secreting stuff from the cell, whereas in the apocrine, we're trying to start to divide the cell, but we don't actually divide the cell. We simply pinch off bits and pieces of the cell and allow that bits and piece to go ahead and be secreted. Whereas in the holocrine, we're actually undergoing the cell division, and then we go ahead and disintegrate the cell for secretion. So we'll take a look at all of these epithelial tissues histologically via slide images and via the slides when we get into lab exercises.